Okay, so I left off a little bit from section 3 that I'm going to review right now. There are 3 key concepts that govern the regulation of digestive activity. So the first one is going to be mechanical and chemical stimuli. So we have receptors located in the walls of the organs that make up our gastrointestinal tract, and these receptors can respond to a variety of stimuli. One of which could be stretching of the organ itself. For example, if we have food in our lumen. Also changes in our osmolarity, which would represent changes in solute concentrations, as well as the pH of whatever is in the lumen itself. The effectors of the digestive activity are the smooth muscle and the glands itself. So when they're stimulated, the receptors in the GI tract can initiate those reflexes that we talked about previously that help stimulate the smooth muscle of the GI tract to help mix the contents within the lumen as well as move them along the digestive tract. And we also have neurons, both intrinsic and extrinsic, as well as hormones that control digestive activity. So our intrinsic controls, again, these are going to involve the short reflexes. And if we're talking about extrinsic controls, those are going to involve long reflexes. Additionally, the cells that line our stomach and our small intestine can produce hormones, or some of the cells. And when these cells are stimulated, they can release the hormones or the products to the interstitial fluid in the extracellular space itself. And then the blood and interstitial fluid distribute these hormones to the target or effector cells, either in the same organ, or those hormones can act at different digestive tract organs. Okay, so now moving on to section 4, we're going to talk about the mouth and some of the associated organs. Again, way more information if you were to open up your textbook and read this section. You may need some of this material for lab because a lot of it is gross anatomy and microscopic anatomy of the mouth and its associated organs. But in these slides are what you need to know for lecture. So if we're talking about the mouth, basically that is where our food is being chewed. We're actually doing the mechanical breakdown of food and mixing it with saliva, which does contain enzymes, and we're beginning the process of digestion. The associated organs include our mouth, tongue, salivary glands, as well as our teeth. So if we're talking about our tongue, really we're talking about a muscle. Okay, so we have intrinsic muscles and we have extrinsic muscles. The basic difference is intrinsic muscles are not attached to bone, and our extrinsic muscles are. So intrinsic muscles help change the shape of the tongue, and our extrinsic muscles help alter to the tongue's position. The tongue does have its own functions itself. So this is going to include gripping, repositioning, and the mixing of food during churning. Once we are breaking down that food and then we are mixing it with saliva, we form what is called a bolus, which is just basically a compact mass of food and saliva. And initiation of swallowing, speech, as well as taste. Okay, our salivary glands, these are going to secrete saliva. What is the function of saliva? It's going to help cleanse our mouth. It's also going to help dissolve some of the food chemicals that we have because there are enzymes within our saliva itself. It helps moisten food and helps compact the food into that bolus that we talked about. One important enzyme that's within saliva is amylase. And if we're talking about amylase, that's the enzyme responsible for digestion of starchy food, so carbohydrates. So we do actually begin digestion in our mouth with the enzyme that we find in saliva. However, we don't begin digestion of all of our macromolecules in the mouth with saliva, only starchy foods, only carbohydrates. We're not talking about proteins or lipids being broken down in our mouth. Most of the saliva that is produced by our major extrinsic salivary glands, meaning that they're located outside of the oral cavity. Some saliva can be produced by our intrinsic salivary glands that are scattered throughout our oral cavity, but not huge amounts. Most of them come from the outside. So we have three major salivary glands. You'll have to probably learn these locations in lab. The first one's your parotid gland. We also have our submandibular gland and our sublingual gland. So this is just giving you an overview of the type of salivary glands or the cells that they're composed of. We have two main types. We have serous cells. Serous cells are going to produce the watery secretion along with enzymes, ions, and a bit of mucin. Our mucus cells produce mucus. So our parotid and submandibular glands contain mostly serous cells, but the sublingual gland consists mostly of mucus cells. So mucus production is going to be the primary goal of our sublingual glands. Composition of saliva. So the majority of saliva is water. So anywhere from 97 to 99.5%. So we consider saliva to be hypoosmotic. Its osmolarity depends on which of the glands are active and the stimulus that's being initiated for salivation itself. Typically, saliva is slightly acidic, so it has a pH between 6.75 to 7, but the pH may vary depending on our diet. So some of the things that are included in saliva or some of the solids would be electrolytes, which are just ions. We also have the digestive enzyme that we talked about before, so salivary amylase. We also have lingual lipase, but I don't really consider that a digestive enzyme in our saliva because it only makes a minor contribution to our overall fat digestion. Really, when we're talking about the enzyme that's breaking down our macromolecules in our mouth or our saliva, we're focused on salivary amylase for starch or carbohydrates. We also have some proteins like mucin, lysozyme, and IgA, as well as waste products like urea and uric acid. There are a couple things that do help us protect against microorganisms. So IgA that I mentioned before, those antibodies, and the lysozyme is really important because it's a bactericidal enzyme. Bactericidal means that it inhibits bacterial growth in our mouth, and it can also help prevent tooth decay. And then defensins we also have, which is not listed here, but basically they function as cytokines and help call our defensive cells, so things like our neutrophils or our lymphocytes, just in case we need them. So how do we control salivation? Those minor salivary glands that I mentioned before, those secrete saliva continuously in very small amounts that are just sufficient to help keep our mouth moist. However, when we eat and food enters the mouth, the major glands are then going to be activated and we have huge amounts of saliva that are going to come in. So much so that our average output of saliva is about 1,500 milliliters per day, but can be even higher than that when our salivary glands are signaled or stimulated. When we're talking about salivation, that's primarily controlled by our parasympathetic nervous system, and that's again going to be our rest and digest nervous system. When we ingest food, we have different receptors like chemoreceptors and mechanoreceptors that send signals to salivatory nuclei that are in our brainstem, so our pons and our medulla. And then as a result, our parasympathetic nervous system activity is going to increase. I'm not worried that you know the specific nerves like your facial nerve, which is cranial nerve number 7, or your glossopharyngeal nerve, cranial nerve number 9. Just kind of understand the signals sent from the receptors to the salivatory nuclei that reside in our brainstem itself. So if we have strong sympathetic stimulation or even dehydration, that can inhibit salivation and result in dry mouth. We can also have just extrinsic stimuli like smell or the sight of food, or an upset GI can also act as a stimuli. Okay, moving on to teeth. In lab, you'll have to know a lot more about teeth, but here, what are teeth? They lie in sockets in the gum covered by margins of the mandible and maxilla. If we're talking about mastication, that's the process of chewing. We accomplish that by our teeth. Dentition and dental formula. Again, you don't need to know how many baby teeth or permanent teeth we have. For lab, you most likely will. Again, here, the dentition and the dental formula, you don't need to know any of this or classification. This is primarily for lab.